Okay, students, welcome back. I'm audible to you all.
Yes, sir.
Okay, so today you got a news that you are coming to the campus from next week. So some of the time that we are having, so in that time we can conduct some of our experiments that are remaining. So many of that we have done online way. So we'll try to do those also as much as possible, and those are remaining, so that we can also conduct in the your face-to-face classes. So all of you are ready to come to the campus? So all of you are ready to come to the campus?
No, sir.
Who is the no? Who is for no? Yuvraj. So you don't want to come to campus. Why you want to come to the campus?
No, sir, it wasn't me.
Okay. So you are coming to the campus?
Sir, we have no choice.
Yeah, you should come to the campus. So now the days are over taking class while in the bathroom or on the bed. Now sit in the class and have classes. Okay. And what's about Vivek? 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 Vivek, I think he is sleeping. So I don't think other than Vivek may have any issue because Vivek might be in States, so he may require some extra time. But other than them, all of you are nearby area only, so you can come easily. Uh, okay, so just as a part of our laboratory session, what we have done, just okay, we have done vast vacuum also, we have done isolation done, isolation, electroporation, restitutionization. Okay, so next is the ligation that we have to do. Okay, so in the ligation, all of we have talked about the first module, the ligase enzyme, what this really ligation is going to be. So before going into methodology, how you have to perform this thing, I'm showing you just a short video, and that short video is going to be able to explain how really this ligation is going to happen, what the role of this enzyme is going to be. It's a very short video, so just are you able to see my screen? Are you?
Yes.
Are you able to see my screen?
Yes, sir.
Yes, sir.
Namrita. Namrita.
Yes, sir.
Okay, hope you are able to see it. So, okay, so I'm playing it.
DNA ligation is the formation of a covalent bond between adjacent DNA fragments, frequently a vector and a gene of interest. Ligation is usually the final step before transformation in the cloning workflow. DNA ligase may be used to join double-stranded DNA fragments with either blunt or
Am I
cohesive
audible?
ends to form recombinant DNA plasmids. A DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in duplex DNA.
Wait for a moment.
Ligation proceeds in 3 steps.
Am I audible now?
Sir, you are audible.
Okay, so
Yes, sir.
was I audible earlier also?
Yes, sir.
And audio of video was also available to you?
Yes, sir. Yes, sir. Yes, sir.
Okay, so now I'm playing the video again.
DNA ligation is the formation of a covalent bond between adjacent DNA fragments,
Simply, this is
frequently a
I'm
vector
that
and
this
a gene of
process
interest.
basically in but
Ligation is usually the final step before
I
transformation
get
in the
joining.
cloning workflow.
This is going to single first and
DNA ligase may be
single
used to
blunt
join
and
double-stranded DNA fragments with either blunt or cohesive ends to form recombinant DNA plasmids.
active ends like it better.
A DNA ligase catalyzes the formation of a phosphodiester bond between juxtaposed 5' phosphate and 3' hydroxyl termini in duplex DNA. Ligation proceeds in 3 steps. Initially, the ligase is self-adenylated by reaction with free ATP, or NAD. Next, the adenyl group is transferred to the 5' phosphorylated end of the donor strand. Lastly, the formation of the phosphodiester bond proceeds through the reaction of the adenylated donor end with the adjacent 3' hydroxyl acceptor, accompanied by the release of AMP. While T4DNA ligase is the most commonly used ligase for cloning, there are several options from NEB to choose from, including convenient master mix formats. For help with selection of a DNA ligase optimized for a specific end type and application, view our selection chart at nebsticktogether.com. Visit clonewithneb.com.
Okay, so I hope that you may be able to get what this DNA ligation is going to be, and the same thing I also have explained you while I'm describing the ligase enzyme. So what are going to be the requirements? So what are the things you have seen in this video? Like you need a ligase enzyme that is going to help in the ligation. And second thing is energy you need, energy being provided by the ATP. So your reaction mix generally what we are going to have, either going to be a buffer, you are going to have with your ATP, etc., or your enzyme might be linked with the ATP that you are going to use. Okay, so now just I'd like to show you what how you have to perform your experiment. So this is the just background wise, and I also share you that. So this is the process that mainly that what we are doing and what we have talked about. Like in the step how we are moving down. In the first step, what we did in the first step we isolate our DNA. Then the isolation of a DNA we go for the your electrophoresis. Then we see what type of quality of DNA is going to be. Then after the electrophoresis, after knowing the DNA, then we go for the isolation of plasmid. So the genomic DNA you are having, plasmid DNA you are going to have. So plasmid DNA is going to serve as a vector, genomic DNA is going to serve as a source of gene. Then after that we talked about restriction endonucleases. So restriction endonucleases job is to cutting down. So with that you can cut your vector also, you can cut your genomic DNA. So that they are going to serve you as a job to provide the insert that you want to insert in your vector. So after getting the both thing, now you are having you have to add the join those your vector with your insert. So now the next step when you are having the vector and having the insert, so next job is going to be the ligation. You have to ligate those things. You have to load your insert over your vector. Okay, so this is the aim. That's why we are moving into this step by step. So once you go with the ligation, then next aim is going to be your transformation. So what are the steps we are referring and transformation, and before transformation you need some cells also, the host cells to make them competent for the transformation. So what are the steps that we are referring? What are the whole your laboratory that I'm describing? This all is the part of molecular cloning. So what molecular cloning is? Just cloning is simply you can define that cloning is a cloning a segment of gene into loading after loading or a vector into a host organism. Okay, so it is the final step in the construction of recombinant plasmid in connecting the insert DNA into compatible digestive vector backbone. So one more thing here you need to focus that when you are ligating the your vector and your insert both must be compatible in terms of ends and in terms of the sticky ends. If you are having blunt ends, then issue is going to be different, then everything is compatible. But if you are having a sticky ends, then only those ends which are having the complementary overhang, they are only able to join. This is accomplished by covalently connecting sugar backbone of two fragments. The reaction is called ligation. So this reaction in which you are joining two fragments is called ligation. And the enzyme that even have shown in the video, and also in our first module we talked about what ligase, from where we are going to get the ligase. So here we are going to get T4 DNA ligase. So T4 is going to be a bacteriophage. So we are having a virus oriented ligase. And this T4 DNA ligase is going to help the phage into the inserting inside the host genome. So the DNA ligase catalyze the formation of covalent phosphodiester linkage. So the aim of the ligation that is going to do is creating a phosphodiester linkage, and the phosphodiester linkage means you are linking phosphate group that is going to be present at the 5 prime and OH group that is present at the 3 prime. So this is the prerequisite that must be there if you want to conduct any ligation. There must be a 3 prime OH and must be a 5 prime phosphate. Okay, so once the ligation is done, insert physically attached to the backbone and complete plasmid. Now this is a complete plasmid. So this complete plasmid you can transform into a bacterial cell for the propagation. So this is a complete vector that you are transforming into the now. Okay, so this is the simply you can say now whatever we are talking about, they are the steps. Okay, so then the majority of ligation reaction involve DNA fragments that have generated by restriction enzyme digestion, because this sticky end generation is making your ligation a bit faster and more efficient. It's not like that if you are having blunt end, your DNA ligase won't join. Your DNA ligase even can join the blunt end, but it is going to be a bit slower process, because it's necessary that two ends should come close to each other. If they are roaming freely, they are very far away, then they won't join together. Okay. Uh, so the most restricting enzyme digests asymmetrically across the recognition sequence. which result in the single-stranded overhang on digested end of DNA fragment. The overhang is called sticky ends. So this is all we have talked about in our when you're talking about your enzymes. And what allows the vector to insert to bind each other when sticky ends are compatible means overhanging base pair happen. And now two pieces are very near to each other and they can be connected. See, two DNA strands, they are come together for just your base pairing, but they stay together or not, you cannot assure because they are not joined. So if you ligate them, then only they are going to stay together. Okay, so because this base pairing, there's a very short overhang is there. So this short overhang, short pairing is not so strong that it will be held together, and just by this base pairing message you can state that now your strand is double, is a circular in nature. Not now, until unless you are not ligating. So it can't happen like you can't rely in a way that just having a sticky end and mix two sticky ends or fragments together and directly transfer. If you transfer that way, then within your uh, host, so host is not going to take care about for the process of ligation. So don't believe that host will ligate for you. No, host won't do it, and host will degrade your single-stranded DNA fragments. Okay, so you are connecting by the ligation. So how it is there? So you can see blue and red two fragments are there. So overhang one and their complementary is there, TT, AA, TT, AA. So they will pair and pairing will combine them and ligation will join them. So what are the reagents you need? So reagent wise, TNAD for ligase you need, 10x DNA ligase buffer you need. So this buffer is going to have your ATP itself. You don't need to add ATP from the outside, or these ligase are going to be associated with the ATP. So it's not like, but ATP is must because from where you will get the energy, or they might be adenylated. So you may say that these ligase are adenylated ligases. What do you need? Linearized vector you need, and DNA fragment you need that you call the insert. So these are going to be a requirement. Then if you talk about what is the method of this ligation, so how you're going to ligate it. So you need to combine all these things in the microcentrifuge tube. And what is going to be your amount, what you need. This is going to be composition of your typical reaction mixture, you can say. So just so that I'm bringing all on a single page. Okay, so this is as in case of restriction digestion, we talk about a reaction mixture. So it is also going to be a reaction mixture that is going to have all the desired component that you need. So first thing, you need a digested vector. And amount wise is a simple thing. If you compare, if you see that amount of insert DNA and amount of your vector DNA, the ratio in between them is going to be 1:5. Vector you are taking in a very large amount as compared to your insert. So why we are doing it? We are doing it so that your insert will get more and more chance to pair with the vector. If the vector availability is more, then the chances that the insert will go and all the insert will be base paired. And when they are going to base paired, that leads to the ligation when you are adding the ligase enzyme to that. So the 50 to 100 nanogram of DNA you need of vector DNA, 10 to 20 nanogram of your insert DNA. Just a minute.
Sir, you are on mute.
Okay, sorry. So the buffer amount, that depends on your reaction mixture. It is going to be 10x buffer. So if your reaction volume is going to be 10 microliter, then you have to add 1 microliter of your buffer. So here also the buffer is a important factor as in case of your restriction enzyme. So this buffer is going to provide you all the condition as you are referring there, necessary conditions, but you are not writing what necessary conditions are. So necessary condition simply means pH and your divalent ions like magnesium ions. So everything is going to be there in your buffer, and that is going to be 10x initial. Then next requirement is going to be DNA for ligase. So ligase is going to be in international units. So international unit means one unit can able to catalyze 1 microgram of your reaction. So just you need 1 unit of your enzyme. So in whatever volume you are, it's there in your solution. So when these DNA ligase generally we are getting from the market. So for that you need to check what your supplier supplied you. The DNA generally it is going to be more than 1 unit per microliter. So depending upon that, you can choose how much microliter of your ligase you need to add. So at least 1 unit we need. So 1 unit per microliter which is there, just add 1 microliter of your ligase. Then the second thing is the last thing that you need distilled water. So distilled water you need to just make up the volume. So like if you want to conduct your reaction, so if your reaction volume is going to be low, then also reaction is going to be very slow. So you must have a reaction volume that significant amount, not a very light diluted, not also very concentrated one. So just generally we perform our experiment in 20-25 microliter. So whatever the composition we have written here, so that we do in 25 microliter of your volume. Then come to the incubation. So when you mix all these things, so when we perform these mixing, we are conducting these at ice. But once you mix all these things, keep back your reagents into the -20. So generally we are storing these enzymes at -20 so that increase its life. And once done, then keep it over ice. So now the incubation times depend upon how long you want to incubate. So if you want to do a quick fast, means if you incubate at 30, as we all know that at 37 degrees is going to be optimum temperature for most of our enzyme. But here, if you don't want to do it fast, if you keep it at room temperature, then just within 20-30 minutes, it is going to do your job. Or if it is the room temperature, so whenever we refer the room temperature doesn't means the temperature we are having. So room temperature generally in the summer, you will say that the room temperature is going to be 40 degrees Celsius. Then we have to incubate at 40 degrees Celsius. Also is no. Wherever we are referring room temperature, we are referring our air conditioned room. And in our air conditioned room, we are generally having our temperature 24-25 degrees Celsius. So where it is written room temperature, that means we are referring 25 degrees Celsius. Okay. So if you keep at 25 degrees Celsius temperature, means our air conditioned room. If you keep it for 2 hours, then your ligation will be done. And if you don't want, if like you perform your experiment in the evening, then you have to keep at 16 degrees Celsius overnight. So now you may ask, sir, so why so different type of thing? When our room temperature is doing a very quick job, then why you are asking us to incubate it so long? So thing is that incubation we are doing so long because all this experiment, all this joining is going to be on the basis of the probability. Probability of what? Probability of two chimeric segment, means one insert that you are saying, and second is going to be your vector. Is these two must come near to each other. But when it's like the short time is if all are coming to near to each other, then that short time is very much enough to join. But it generally rarely happens that as you edit the enzyme, insert and vector, they come close to each other and joining done. Okay. So to increase the efficiency so that whatever the insert is there and that all the insert got ligated, we increase our time. But then you may argue that, sir, why then we are incubating our time 37 degrees Celsius or room temperature for longer duration? That is because your enzyme stability. So if you keep longer time on your room temperature or 25 degrees Celsius, the stability of enzyme got sacrificed. So enzyme no more remain very much functional. So that's why we are keeping a bit lower temperature, maintaining the functionality of your enzyme and also increasing the efficiency of your joint. So once you've done it, once your incubation is over, the next thing is to check to confirm whether ligation is done or not. Because still we can't say just once we added the ligase enzyme and the ligation is definitely going to be over. No, it may not happen. To confirm whether the ligation is done or not, so two things are there with us. One thing is agrogel electrophoresis. So you can check on the agrogel electrophoresis, and you know that earlier the vector was linear. Now it is going to become circular. So once it's going to become circular, then it have to show all its that three different type of the conformation that we are referring. And if there is no ligation, then it's going to be linear, and the size is going to be as size of your linear DNA. So you can compare there also whether your ligation is going to be there or not. But one drawback is there in that case, like when it is going to be circular one, and circular one is running at three different sites, linear one at a different one, and nick one and supercoiled at different one. So in that way we cannot clearly say whether the ligation happen or not, or if very few ligation, very few, it's not even not efficiently ligated. But if you feel that one or two fragments they got ligated, or 5 fragments got ligated, so 95% not ligated, 5% got ligated. In principle, we need one. Even if the one strand got ligated, that's enough for us. But as much like got ligated, that is going to be beneficial for us. So to confirm that at least some ligation is going to be there, so the gold standard way is go for the transformation. If the fragments got ligated, and if you transform into the host, like E. coli is a host, if you transform it into the host, means if you make host to pick up it. So transformation is a process in which you transfer a foreign DNA into a cell. The process of transfer of a foreign DNA into a cell is called transformation. Okay. So if you transfer your host vector into, or your recombinant vector, recombinant means it is a vector itself plus your insert into a cell, if the ligation is done, then plasmid have ability to establish itself, means it have origin of replication. So as it go inside, it will start replicating within there, and so it will start. Once it start replicating, then now it's your job is done, and you can screen, you can check means that your screening process that we all know that the markers are there over your plasmid. So on the basis of the marker that are there on your plasmid, you can check whether the whether the vector is there or not. But here also one more issue is there. You want to check whether it's your vector ligated itself. So the possibility is there, vector may self-ligate. Without insert, like this is a vector. Like this is a vector, you can say it's open one, and in this vector you have to insert this event to insert. So when the insert is going to be there, your vector should be like this. But sometimes chances are there that it's very likely that vector got self-ligated without the insert. So when such type things are going to be there without the insert, vector is going to be ligated. So how to check it? So you have to check on the basis of insertional inactivation. Your vector is going to have markers. So markers on the basis of even empty vector is there. That also give you the activity of resistance. Now double markers that you're using, like two antibiotic resistance are going to be there. So one antibiotic resistance is going to be inactivated. So if this insertion is going to be there, that leads to the insertional inactivation. So that insertional inactivation you can check by growing on your two different antibiotic plates. But one more type of screening is there that the blue-white screening is there. Now the single way, now you don't need to go for a replica plating type of things. That also you can go. If the insert is there, then colonies are going to be white. If there is no insert, then the colonies are going to be blue. So this blue-white screening you can go with. This is how you perform your ligation. What precaution you need to take care? Because ligase buffer contain ATP. So this is a thing. That's why I was referring you here the need of the temperature that we are referring. Because ligase buffer contain ATP, which degrade upon freeze-thaw cycle. Means we are quickly freezing, thawing, means bringing to the -20, coming back to the your room temperature. That is going to degrade your ATP. And if it got degrade, then it won't be functional. So in that case, what to do? You should aliquot your buffer in a way that each and every time you need not to take out the stock vial, you can take out the vial that you need and use that. That individual aliquot of 5 microliter, 10 microliter, 20 microliter you can keep, and you can store individual aliquot into your -20. So whenever you need, take out one aliquot and use it. Whenever you need setup ligation in the future, you can throw new tubes and you can proceed with it. Okay. So once and like 5 microliter is there, so you need only 2 microliter. So once, twice, thrice it can be okay. So but longer duration, frequently, daily you need, daily you are thawing, then it's going to degrade your ATPs. And control must be there. So control means vector alone. So if the vector alone is there, so what type of things are going to be there? And if no ligase is there, if you are not adding the ligase, so then what type of results you are getting? So all these types of control you must keep. So they are going to check whether the ligase is functioning or not. And DNA should be properly purified. So purified DNA is must, because if DNA is not purified, then it is going to be it will interfere the ligation process. So this is the experiment that we do, and so now I'm giving you a 5-minute break to just read it again, and if any question from this theoretical part if you're having, you can ask, and then we'll move for attendance, and after that we'll go for our next session. Okay.
Excuse me, sir.
हाँ जी बोलिए.
So would it be fine if you make all of us attendee? Actually, someone is keep on removing us. Many of the student has removed during the meeting.
Yes, so just wait for a moment. Okay, so I think all done. So all attending now. Okay, students. So now we can end our session. So we'll join back after 5 minutes. In that we will discuss some more aspect of, or some we'll see some more videos. So how really in our lab we are going to perform this experiment. Okay. So see you again after a 5 minute break.
Sir, you're not audible.
Vivek. Vivek,
Yes, sir.
you want to say something?
Sorry, sir. No, actually, in between, your voice is not audible. That's all I
मैं
wanted to.
आपको आवाज लगा रहा था, आपने बोला नहीं शुरुआत में। आ रहे हैं आप मंडे से?
आप बुला रहे हो सर, तो आ जाएंगे.
नहीं, हम तो बुला रहे हैं, आप ही लोग नहीं आ रहे।
नहीं.
तो आप यही हैं या स्टेट्स में हैं?
सॉरी सर.
आप भारत में हैं या संयुक्त राज्य अमेरिका में हैं?
No, sir. I am in America right now, but I'll come back if it's mandatory.
Yeah, now it's mandatory, so it's mandatory for all.
Okay, sir.
Okay, but if you have to come from the America, then you can have some more day concession. So for your tickets, etc. For that, you have to write to the HOI, or you have to write to your PL and CC to HOI, or write to HOI CC to your PL. Okay. So giving the details that when you are booking, what your itinerary is. Okay.
Sir, someone keeps turning my audio off.
See, no one can turn on your audio. Only your audio can be turned off.
I don't know, sir. It's turning off constantly. Like even now it just turned off. I'm not able to hear you.
I don't think it might be some error in the team. That's why it's happening.
Okay, sir. Okay.
Otherwise, it should not happen. Yes, you can be muted. Like Yuvraj can do, I think. If your Yuvraj is doing, because he was in the presenters list, so he can do. Yuvraj, Yuvraj, Yuvraj. So युवराज करके भाग गया, देखा भाई? तुम क्यों परेशान कर रहे हो? Okay, so we'll join back after 5 minutes. Please all of you turn on your camera for recording your attendance.